DNA replication, copying the blueprint of life. Definition: DNA replication is the biological process by which a cell makes an exact copy of its DNA before cell division. It ensures that each daughter cell receives a complete and identical set of genetic material. Nature of replication: DNA replication is semi-conservative. Each new DNA molecule contains one original, parental, strand and one newly synthesized strand. Bidirectional replication proceeds in both directions from the origin. Semi-discontinuous: one strand, leading, is synthesized continuously. The other, lagging, in fragments, where and when it occurs in eukaryotes. During the S phase of the cell cycle, inside the nucleus in prokaryotes. In the cytoplasm, begins at a single origin. Key components: component role, origin of replication, ORI. Starting point for replication: helicase unwinds the double helix single-stranded binding proteins, SSBPs. Prevents strands from re-annealing. Topoisomerase, e.g., DNA gyrase. Relieves supercoiling. Primase synthesizes short RNA primers. DNA polymerase adds nucleotides to the growing strand. Sliding clamp holds DNA polymerase in place. Ligase joins Okazaki fragments on the lagging strand. Nucleases remove RNA primer. Steps of DNA replication. 1. Initiation begins at the origin of replication. Helicase unwinds the DNA helix, creating a replication fork. SSBPs stabilize unwound strands. Primase lays down short RNA primers. 2. Elongation. DNA polymerase III, in prokaryotes, adds nucleotides to the 3' end of the RNA primer. Letting strand is synthesized continuously in the 5' right pointing arrow 3' direction. Lagging strand is synthesized discontinuously as Okazaki fragments. DNA polymerase I replaces RNA primers with DNA. Ligase seals the gaps between Okazaki fragments. 3. Termination. Replication ends when the entire DNA is copied. Telomerase extends the ends, telomeres, of eukaryotic chromosomes to prevent information loss. Leading versus lagging strand feature: leading strand, lagging strand. Direction toward replication fork, away from replication fork. Synthesis: continuous, discontinuous. Primer needed. 1. Multiple fragments. No. Yes. Okazaki fragments. Replication in prokaryotes versus eukaryotes feature: prokaryotes, eukaryotes. Origin of replication: single, multiple. Speed: faster, slower. DNA polymerases: fewer types, multiple types, alpha, delta, epsilon. Telomeres absent, present. Location: cytoplasm, nucleus. Importance of DNA replication: maintains genetic continuity across generations of cells, ensures genomic stability critical for growth, development, and repair. Errors may lead to mutations, cancer, or genetic disorders. Proofreading and error correction: DNA polymerase has 3' right pointing arrow 5' exonuclease activity. For proofreading, mismatched bases are detected and corrected. Additional repair mechanisms include mismatch repair and nucleotide excision repair. Inhibitors of DNA replication used in research or therapy. AZT, zidovudine, inhibits reverse transcription in HIV. Ciprofloxacin inhibits DNA gyrase in bacteria. Hydroxyurea inhibits ribonucleotide reductase. Conclusion: DNA replication is a precise and tightly regulated process that ensures each new cell receives a faithful copy of the genome. It involves a coordinated effort of many enzymes and structural proteins, and its high fidelity is essential for life.